In this video, I'm going to go over some of the main points that we've covered thus far in this unit. So, one of the first things you really need to understand is the hierarchy of life. You need to understand that we are organisms. A lot of organisms are made up of organ systems that all function together to help the organism perform its life processes. Those organ systems are made up of organs. The organs are made up of tissues, which are groups of cells that perform a specific function. And those tissues are made up of cells. The cell has organelles inside of it that perform specific functions. Those are made up of macromolecules. And along with that, there are macromolecules that also perform specific functions. So we went a little bit in depth into cell organelles. You needed to review their location in the cell, what they looked like, and their function. So make sure you take some time to review this image, review the function, especially of the nucleus, the rough and smooth endoplasmic reticulum, the cytoplasm, the mitochondrion, the membrane, the Golgi complex or Golgi apparatus or Golgi body, and review the differences between animal and plant cells as well. Then you learned about the four macromolecules. The four macromolecules are carbohydrates, nucleic acids, proteins, and lipids. We're not really going to go into too much depth with carbohydrates. You learned that sugars are made through photosynthesis. So the monomer of carbohydrates is sugar. And the polymer, when you link a bunch of sugars together, you get starch. Nucleic acids we're really going to go in depth with here in this unit. So a chromosome is made up of DNA, and DNA is made up of nucleotides. So the monomer for nucleic acids are the nucleotide. And then the polymer in this example is a DNA strand, but you'll also learn that nucleotides can make up RNA as well. Then we have proteins, protein macromolecule. The monomer of a protein is the amino acid. When you connect a bunch of amino acids together, you get a polypeptide or a protein. And finally, we have lipids. The monomer is considered the fatty acid, and that is a long chain of carbons surrounded by hydrogens. You put a few of these together and you'll get the polymer of lipid. So focusing in specifically on nucleic acids in this unit, again, the monomer of nucleic acid is the nucleotide. The nucleotide has three parts, and that is a phosphate, a sugar, and a nitrogenous base. When you connect a bunch of these nucleotides together, you get the polymer, which will in this case be DNA, but it could also be RNA. So a couple of differences between DNA and RNA. So obviously the first difference is the name. So DNA and RNA are both named after their sugars. So you see here that the sugar in DNA is deoxyribose, hence the name deoxyribonucleic acid, DNA. And for ribonucleic acid, RNA, the sugar is ribose. So we have the name ribonucleic acid. So that's the first main difference between DNA and RNA. And I'll go into a little bit more detail in a couple of minutes. So the general structure of DNA is the double helix. So that's just two strands of nucleotides twisted together. So it kind of looks like a twisted ladder. That's the double helix. If we untwist it and examine it a little closer, we see the chains of nucleotides bound together to make up the strand of DNA on both sides. So two strands. One part of the structure is the backbone. So this is one backbone, and then here is another backbone. But what you'll notice is that one runs one way, the other runs the other, and that's why they are anti-parallel. The rungs of the ladder, well, let me go back to backbone for a second. Notice that the backbone is made up of a sugar and a phosphate all the way down. Sugar, phosphate, sugar, phosphate, sugar, phosphate, both sides. Sugar, phosphate, sugar, phosphate, sugar, phosphate. So the backbone is made up of sugar and phosphate. The rungs of the ladder are made up of the base pairs of the nucleotide. So we have one base here, one nitrogenous base here, one nitrogenous base here. One here, one here. Okay, so the rungs of what we would visualize as the ladder are made up of the nitrogenous bases paired together. DNA replication is pretty straightforward. The first thing that needs to happen is the DNA double helix needs to unwind and separate, and DNA helicase is a protein that does that. Once it's unwound, free nucleotides floating around inside the nucleus will be attracted to the opposite strand, and always A will be attracted to T, which adenine is attracted to thymine, and cytosine is attracted to guanine. So once they're close, DNA polymerase on both sides will bind them together, and you'll end up with two identical copies of DNA, hence DNA replication. But what's really important to remember is adenine always binds to thymine and vice versa, thymine to adenine, and cytosine always binds to guanine and vice versa, guanine always binds to cytosine. So back to some differences between DNA and RNA. So I talked about the sugar. RNA uses ribose instead of deoxyribose. Okay. The sugars are just very, very slightly different, but because they're different, they have different names. The second difference is that RNA will use uracil in place of thymine. There is no thymine in RNA, and I'll show that in a minute. But uracil is used instead of thymine. There's no thymine in RNA. So one way to identify an RNA strand versus a DNA strand, if it has thymine, it's DNA. If it has uracil, it's RNA. And the last final difference that I need you to know is that RNA is typically single-stranded and DNA is double-stranded. So we're going to get into transcription and translation over the next few days. So you need to understand how RNA binds to DNA, or how the RNA strand is made from the DNA. So as I was saying before, where you would typically find a thymine binding to an adenine in DNA, in RNA you're going to find a uracil binding to an adenine. So any place where thymine would have been for DNA, it's now going to be uracil. So adenine still binds to thymine, but again, where you would have had thymine in the DNA, you're now going to have uracil.